Hello and welcome to our webinar titled Towards Real-Time Release Testing, Dissolution Prediction of Tablets Made Using Continuous Direct Compression, hosted by Biopharma Asia Magazine and presented by Dr. Pallavi Pawar. My name is Sri Patel and I will be the moderator. Now, before we begin, I'd just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we'll go through these at the end of the presentation. Now, please allow me to introduce you to our presenter. Dr. Pallavi Pawar has been working as an investigator in pharma R&D in the product and process engineering team at GlaxoSmithKline since September 2016. She supports the progression of GSK's new chemical entity portfolio by developing robust continuous formulation processes using state-of-the-art technology, modeling, and statistical experimental design techniques. She works primarily in R&D labs, and as project advances through development, she is responsible for scale-up, culminating in tech transfer to commercial manufacturing facilities. She received her PhD in chemical engineering from Rutgers University in May 2016 under the guidance of Dr. Fernando Muzio. Her thesis focused on tools for real-time release testing in batch and continuous tablet manufacturing. In it, she investigated non-destructive methods like near-IR spectroscopy to predict process and product performance, blend and content uniformity, hardness and dissolution of tablets, and compared this approach to traditional testing in both batch and continuous manufacturing mode. Her expertise is in powder handling, material characterization, and powder processing geared towards direct compaction of tablets. She has worked with batch and continuous unit operations, including powder mixing, powder feeding, twin screw granulation, fluid bed drying, milling, and tablet compaction. She completed her bachelor's degree in chemical technology from Institute of Chemical Technology, Mumbai in 2010. I would now like to invite our presenter, Dr. Pallavi Pawar, to join our webinar. Welcome, Dr. Pawar.
Thank you, Sri. So as Sri said, I am going to talk about the concept of real-time release testing, especially applied to an important tablet critical quality attribute, that is tablet dissolution. And to further explain this, I am going to jump into a case study that we did at Rutgers University during my grad school, where we applied this concept of real-time release testing for dissolution prediction of tablets made on the continuous manufacturing line. And as mentioned, this work is from my grad school at Rutgers University. So then, let's directly jump into the presentation. One second. Here. Okay, so this is the outline of my talk. I will give you a brief background about real-time release testing and highlight the advantages of RTR. We'll then jump into the regulatory perspective on RTR, and then there's a lot of data out there, there's a lot of guidances out there. I'll just briefly walk through them, but I would highly encourage people to go to those guidance, refer those guidances for further information. So after that, we will focus on RTR in dissolution testing, after which I'll give you a brief background about the tools that we've used in our case study to extract information from our process data. And by those tools, I mean use of near-infrared spectroscopy, and then I'll also talk to you about how we extracted information from the dissolution profiles. And so then we'll jump into the case study. I'll talk about the experimental setup, I'll talk about the materials, the design, the approaches that we have used to investigate and analyze the data, and so on. And then I'll talk about the results that I have for this particular case study. We'll finally conclude it by discussing the validation strategies for real-time release testing, especially for the approach that we used. And after that, I'll conclude my talk and give it back to Sri. So what was the motivation for this work and what is real-time release strategy? So we all know real-time release strategy is the ability to evaluate and ensure quality of intermediates and final product based on process data, material attributes, and in-process testing. So this is the general definition of real-time release testing. And so we all know that different regulatory bodies like the FDA define certain critical quality attributes for intermediates and final product. And so what RTR aims to do is to predict these CQAs during the process and not depend on after the fact assays. And so then what CQAs are we talking about? We are talking about for direct compaction, the CQAs could be your content uniformity of the tablets or the tablet ID, and then eventually dissolution testing. So if you were to look on the figure in the right, I'll give you a quick example of how you can apply RTR strategies in such cases. So for example, RTR for a certain CQA can include gathering information from the process data. Like you can obtain information from the continuous blending operation that you have there. Besides that, you can obtain information in the form of the knowledge of the incoming raw materials. And you can put that together with the information that you obtain from the in-process measurements, in-process point measurements. An example of an in-process measurement is just looking at the concentration and the uniformity data by plugging in NIR in your system, or any other PAT tool for that matter. Or another point measurement could be the information that's obtained from the tablet press, like the compaction source. And you can use all this data for your dissolution model or to predict content uniformity real time. So this is how you can make use of different information that you gather from the process and your raw materials towards real-time release testing. So what are the advantages of real-time release testing? There's an extensive amount of online and at-line information or data that you gather. You interrogate larger number of samples, and which gives you higher assurance of the product quality. You also collect data in process. And so if there are any corrective actions that need to be done, you can implement it real time. And this is very, very important for continuous manufacturing because you cannot reprocess a lot. And that's when you have to acquire information or collect information real time and make changes to the process if needed real time. And so real-time release testing also implies lesser end product testing, which reduces inventory. It lowers laboratory costs. So these are the advantages. Let's go ahead and then look at the regulatory perspective on RTR. So this approach was, the RTR, real-time release testing approach, was highly encouraged by the FDA. And this is evident from the FDA's PAT guidance of 2004 and also the ICH guidelines. So the guidance of 2004, the PAT guidance of 2004, it has some language about RTR. And as suggested, this is just a quick overview of the strategy or the regulatory perspective on RTR. I would encourage everyone to go back and look at these different guidances, which are very, which help and which discuss a lot of things on RTR. And so what the PAT guidance of 2004 says is you need to have process understanding, you need to have sound control strategies in place, and you can couple that with online, inline, or at-line measurements so that you can successfully implement RTR. There is another working group, the ICH Quality Working Group, which published this document, a question and answer document in 2010. And I've just picked a few question and answers here, but there are lots of question and answers there which discuss real-time release testing strategy. And so for example, they talk about if you can use RTR to make batch release decisions and to avoid end product testing. And the answer there is, if you can predict all CQAs and hence the batch quality is assured by real-time process monitoring, by the knowledge of material attributes, and as I mentioned earlier, by inline, online, or at-line testing of materials, then you might be able to release your batch based on that data for that particular CQA, and you can avoid end product testing for that particular attribute. The one thing that you need to remember to apply this approach is you have to justify the sampling strategies. You cannot rely on traditional sampling strategies. You need to have a very sound sampling plan in place. The last point is something that's interesting for me. They talk about using surrogate measurements for RTR. And by that, what we say here is, can we use process parameters and material attributes to predict a certain CQA? And their answer is yes, we can do that. You can use surrogate measurements like process parameters and material attributes that have been demonstrated to correlate with your in-process and end product specifications. And an example of this is, say you're looking at your content uniformity model. You can build your content uniformity model based on surrogate measurements, like you can, like process parameters. An example process parameter is the data that's collected from the feeders in the continuous manufacturing line. So you can use measurements like those. You can use incoming material attributes. So you have to factor that in for sure. And one such attribute could be the particle size distribution or the D50 of your API. And then you can also rely on the in-process measurements like the tablet property, for example, where you're looking at the tablet thickness and weight. So all these surrogate measurements together can help you predict or build an RTR model for content uniformity. So with that, let us now move on to RTR for dissolution and see how we can apply RTR for dissolution testing. So we need dissolution testing for the development, registration, approval, and quality control of tablets. However, dissolution testing is a destructive, time-consuming, expensive method, and it requires media preparation, and it's absolutely useless for online quality control. And to add to that, it interrogates only a limited number of tablets, which can decrease our confidence in the batch. And so we need a tool that's non-destructive, that's fast for prediction of dissolution. So now what do we need to consider to build RTR models for dissolution? We know that dissolution takes several minutes to hours. And there can be two different approaches to predict dissolution. In case of immediate release tablets, if you are only interested in predicting the Q point, that is the drug released at a certain time point, then you're well off. You can just quickly use your model and use the information only for that particular Q point. However, if you are interested in predicting the entire dissolution profiles, which is the case most of the times, then you have to use a different approach. And why do you need to predict the entire dissolution profiles? It's interesting because if the objective is to predict the rate of the drug release or to monitor the potential changes in the behavior of tablet dissolution, then you might be more interested in predicting the entire dissolution profiles instead of predicting the amount of the drug released at a certain time point. And so in such cases, there's no sensing interface that's available to predict the entire dissolution profile directly. And that's where you have to then make use of mechanistic models, and then you can combine it with PAT tools to predict dissolution. And that is exactly what we have done in our work. We've used PAT tools, and in our case, we used NIR. And for extracting information from the dissolution data, we used mechanistic models. And I'm going to talk about both these approaches. The first is how to extract information from NIR. And then I'm also going to talk about how we went ahead and extracted information from the dissolution data. And this is just so that I'm preparing you, because I'm going to talk about the case study where we used both these methodologies. And so as mentioned earlier, For applying RTR, we require tools that can gather data fast and they can gather data real time. And near-infrared spectroscopy is one such tool. So NIR can be used to obtain process information as well as product information. And so when you have to, so if you scan your tablet using near-IR spectroscopy, then what remains is to extract information, which can be of qualitative information or it can be quantitative information. And you need to extract information from the NIR spectral data. And you can do that with the help of different algorithms that are mentioned here. You can use principal component analysis, you can use partial least squares or different regression methodology. So if you extract information using principal component analysis, for example, and you can use it to relate to your responses using any kind of regression methodology. So in the figure that you see at the end on the slide, we have spectral data on the left. This is the data that you are going to get from your NIR spectrometer. This data is multivariate, and that's when to extract just the main information from this, you can use algorithms like the PCA. And I'm going to talk about that on the next slide. So what is PCA and how does it exactly work? PCA is a data compression algorithm which allows you to extract systematic variation in the data. So you can use PCA algorithm on any data set. It does not have to be only spectral data. You can use it on any other different kind of data as well. So now you imagine you have this data set in two-dimensional space. And so what PCA does is it will look at the variation or the major source of variation and the direction of that major source of variation. And you see here that the major source of variation or the direction is captured using the principal component 1. And so what it says is the principal component 1 or the first PC captures the maximum variation in the data. And so now what you have to do is treat this PC1 as your new axis, and the position of the data points with respect to this axis is then determined. And so how you do this is by looking at the residual, which is nothing but the distance or the residual, the remaining or the unexplained variation. And you look at this something called score, which is just the new x and y for this data point using on the new coordinate axis. And basically for the given data point, the green data point that you see here, score is nothing but the projection of this data point from the original x and y space onto this new principal component 1 space. And so it just signifies the intensity of each of the compressed variables for all the samples. So you repeat that activity for each of the data point that's here. So for each data point, you are going to get a new score. You find the next principal component, which is going to explain the remainder variation in the data set. So we see there is more variation along an orthogonal axis right now, and so PC2 is the sequential principal component that explains the remaining variance. And so you do similar exercise for the data points here, where you try and find out the scores for the PC2 axis now. Okay, so this is the way you treat or extract information from the NIR data. How do you extract information from the dissolution profiles? And you have a variety of ways to do that, and a lot of you must be aware of it. What you typically do is you if you're interested in the Q point, then please go ahead and do that directly. But if you are interested in predicting the entire dissolution profile, you can do it in multiple ways, and I have just 2 ways here on the screen. You have a model dependent approach, or you can have a model independent approach. For model dependent approach, what you typically do is you have a huge data library that has different models that you can fit to your dissolution data. And what it gives is you can extract, once you complete that exercise, you can extract the parameters, in this case the alpha and beta that you see on the screen here on the left. you can extract those parameters for that particular dissolution profile. And so you repeat this exercise for each dissolution profile that you have. So per dissolution profile, you're going to get a set of parameters that describe the shape and the level of that particular dissolution profile. So that's one approach. Now imagine you have a dissolution profile for which you are not able to find any equation that can fit that data set. That's when a colleague of mine was looking at this particular way in which you can fit the data set, and it's called model independent approach. And it's what it does is it looks at the level, or it looks at your dissolution profile in terms of level and shape. And by level is nothing but it's calculated by using the average of the drug released at each of the time points across all the time points for the given graph, for the given dissolution curve. And now for the next shape factor, what you do is you subtract the effect of level from your dissolution data and do a PCA on the remainder matrix. And what it does is it captures the remainder variation in the data, which is nothing but it's proportional to the shape of the data. And so typically what you end up getting is, with such an analysis, for each graph or for each dissolution curve, you get a level value and you get values for shape 1, shape 2, depending on how many principal components you get. And so you can define your dissolution curve in that manner. So with that, now what we are planning to do is we are just planning to use our model dependent approach. But if you go back to the manuscript that I had on my first slide, you can also track the model independent approach that we have applied, and we have shown that it works as well. So with that background, let me jump into the case study that we had. So our intention here was to use mechanistic models combined with near-infrared spectroscopy to predict tablet dissolution in continuous manufacturing. And so this is a schematic of continuous manufacturing line. In continuous manufacturing line, we all know, or for that matter in any manufacturing process, we know that powder is exposed to different unit operations, which alters its properties. And the effects of all these individual processing steps is directly reflected in the properties of the final dosage form, which in our case is tablets. So your tablet is going to carry the information of your processing as well as the formulation information. And so what you need is a tool that can extract all such information. In our case, we used NIR spectroscopy, but there are a lot of PAT tools out there which you can use. It has the capability to capture the effects of changes in both formulation and process parameters. And we have done previous studies, and a lot of you must be aware that NIR can capture the effects of compaction force. It can capture the effects of shear strain, and we have published a paper on that. It is also known to, it's obvious knowledge that it can capture API concentration information as well. So our hypothesis for this work was then to use NIR of tablets combined with this mechanistic approach for data extraction for dissolution, and then use to predict dissolution of unknown tablets. And so for that, we used a process understanding approach, and this is a very general flowchart that you can use. What it says is the most important factor here while you predict or while you use this approach is to understand the process. And so what it says is you have to identify, the first step is to identify the critical material attributes and identify the critical process parameters for the given formulation. You then go ahead and identify the dissolution mechanism for the formulation. And so you understand what is the exact mechanism that governs the dissolution. Is your tablet swelling, or is the tablet surface eroding? You need to find out all those things. And then you need to understand how your CMAs and CPPs affect dissolution. So that's your process understanding. Then you go ahead and you design a DOE around all these CMAs and CPPs. And this is more from the continuous manufacturing standpoint, but you can apply this to a batch approach as well. So the next step is then to run this DOE, collect the tablet samples that you've obtained from the DOE at different conditions, then subject that to NIR spectroscopy, so analyze those tablets using NIR, and then run PCA, extract the scores that I spoke about just recently. So those are just to extract the key principal components, and hence you extract the scores. And then go ahead and dissolve those tablets. And by doing that, you know the dissolution profile and the NIR for each of those tablets. And so you can extract information from the dissolution profiles as well. And what the last step is basically what you build a predictive model where your Y, which is your response, is your dissolution parameter or parameters, and your X or your predictive variable is your scores that you obtain from the NIR spectroscopy. And then the last step is to then predict the critical quality of the CQA that's of interest to you at the target condition. So you can always use this flowchart more or less for different CQAs, and we've used this flowchart for dissolution testing and dissolution prediction. So I'll go to the setup that we had at Rutgers. We had two feeders that fed API and excipients into a mill and then to a blender. So the formulation that we used here was semifine acetaminophen, lactose monohydrate, and magnesium stearate. The two feeders, the API and the excipient feeder, sat on the third level of this mezzanine that you see, which is a state-of-art continuous direct compaction line at Rutgers. These two feeders sit on the top floor, the third floor of this mezzanine. These feed materials into a co-mill, which is used basically to delump these materials, which is then transferred into a blender. It's a tubular blender. It's a tubular continuous blender with 24 blades that we used. So this blender mixes all these materials. Your magnesium stearate, which is your lubricant, is not added in the co-mill to avoid the high shear environment. It's directly added into the blender. So after the blend's made, after the materials are blended along the length of the blender, you then move it to a 36 station tablet press. And so the drill is basically, after you collect the tablets, you first analyze them using NIR and then subject them to dissolution testing. And so this was the experimental design that we had. What we had here was we had three levels of four different parameters. We varied the API concentration, the acetaminophen concentration. We varied the blender speed at 3 levels. We varied the feed frame speed, because we know the feed frame speed is an important process parameter which affects the lubrication lubrication of your blends, and hence affects the dissolution. So we wanted to vary that parameter. And then we also varied the compression force, because we know that affects dissolution as well. And so we used a fractional factorial design for this, which resulted into a total of 27 design points, and then we had 3 center points. So for each of these conditions, the way we ran this work package on the continuous line is we set all the parameters for the first design point. We let the continuous line that I showed in the previous figure, on the previous slide, reach steady state. And once that was done, we collected 6 tablets that were coming out of the tablet press. Then we went ahead and changed our settings, and we went to the next design point, let the system reach steady state, collect 6 tablets, and continue this process. So all these 30 run DOE we were able to do on the commercial scale in a matter of 3 days, or rather 2.5 days. So which resulted into a total of 180 tablets, which were then fed into our calibration model. So once we had this calibration model for NIR and hence dissolution, so we related, so the way we did this was we used NIR spectroscopy on the tablets and then subjected these tablets to dissolution testing. And as I mentioned on the flow chart, we built predictive models which could predict the alpha and the betas which are the parameters for dissolution testing. And so for that, I'll show you the results for this work package. So our first step here was, of course, process understanding. We needed to understand how our CPPs and CMS affected our dissolution profiles. And so we saw here was that there was a very clear-cut effect of compaction force on the amount and the rate of the drug released. We knew that the APAP concentration had to affect the rate and the amount of the drug released. Something interesting that we saw here was the feed frame speed did have quite an effect on the rate of the drug released, where as you increase the speed, it increased the lubrication of your blends, where your magnesium stearate coated on your powders, and it decreased the rate of the drug released. So with all the information here, gathering all this, we then went ahead and looked at the NIR data. So as I told you, I had those 180 tablets. I scanned those for NIR. I scanned them using NIR spectroscopy, and we wanted to see if my NIR was now able to capture all this information. So NIR, we could easily see that it definitely captured the effect of the compaction force, and it's evident here how you have different spectral data being segregated in different areas. We have color coded them for better visualization. So we knew there was an effect of compaction force. We could see that if you pre-process this data, you could see that there was an effect of APAP, changes in the acetaminophen concentrations, and you could see that in the two peaks, one around 8,500 centimeter inverse and one around 11,000 centimeter inverse. And these are two peculiar acetaminophen peaks. So we knew that we captured the information of compaction force and that of concentration using this, using NIR. So we also, then we subjected this data to data compression methods, which is the PCA, and extracted this, what is called the scores. So these are the scores here. We could very well see that your principal component 1, that is, and the principal component 2, combined with the principal component 3, captured 100% of the variation in the data set. We could see here that your PC1 actually explained or captured the information corresponding to the compaction force variation. And to verify this, what we did was we had measured the tablet thickness and hence the porosity of the tablets. So we quickly plotted the scores on PC1 as against the relative density, and we confirmed that my PC1 was capturing the effect of changes in tablet porosity because of this good correlation that we see here. Then we went ahead and looked at the principal component 2, which was very nicely able to capture the variation in the API concentration. And this was also verified by looking at the loadings plot for the NR data, where you see here that the absorbance is highest in these two regions, which corresponds to the API peaks, the acetaminophen peaks. And so we were also able to see that my PC3 captured shear strain. So my next step was, now we know our dissolution, the way our dissolution profiles look. We know how our NIR data looks. The next step was to take these responses alpha and beta, which was extracted from the dissolution profiles by the methodology that I explained earlier, and then use your PC1, PC2, and PC3 scores as your regressor variables. And you build a multilinear regression to predict each of these. So once I have nice predictive equations for my alpha and betas, my next step is then to predict the dissolution profile for an unknown tablet. And so for our case, our unknown tablet contained 9% acetaminophen, 25.4 kilonewtons was the compaction force that was used. The blended speed was 200 RPM and the feed frame was 25 RPM. So the first step there was to project this data onto the spectral space that we had from the scores plot earlier, and also to look at the dissolution profiles. So you see here how we had dissolution profiles for these 6 tablets. And since we had the NIR data for these 6 tablets, which was projected onto the PCA space, we could see very well how these green tablets, which is your target concentration data set, went and clubbed with the likes, with the tablets with similar concentration and similar compaction force. So this is the first step of validating a process, where you can see if the given test set goes and is contained in the region that you want to validate it. And so then we went ahead and predicted the dissolution profiles using the model dependent approach for these 6 tablets. And so you can see here in red is the predicted plot, and in blue is the reference plot of each of these 6 tablets. And so we could see more or less that the predicted dissolution profiles agree very well with the reference plots. And so we just confirmed this using an F1 and F2 analysis, where both the analysis said that all the dissolution profiles were comparable. And so we have done a lot of validation on these tablets as well, which is not shown here. But a general methodology that you need to consider for validation and for batch release of your batch based on your dissolution data is that the first step, it's shown in this flow chart here. The first step is basically to see if your spectra is similar. If your NIR, if you want to use NIR, then you can see if your NIR spectrum for that particular tablet is similar to the target compaction force and strain data. If yes, then you move to the next step. If no, then you directly reject the batch. You also then go ahead and see if your tablets have the right concentration using the NIR as well. If yes, then you go ahead and predict the dissolution profiles. But you do not stop at this point. You then go ahead and see if the predicted dissolution profiles agree with the reference profiles that you have. And then you release the batch. And so once you've some confidence and you've validated this process a few times, then you can use this model going forward to release your batches. And so a few things that you need to consider here while you validate the validation strategy and batch release for dissolution is that you have to have a reference method which is discriminatory. So our dissolution method was discriminatory, where we could detect differences based on compaction forces, and for that matter even ensure that the material experiences in the feed frame. You should be able to reject, your model should be able to reject material that's non-conforming. And I have not shown in our graph previously, I showed you material that was conforming, the tablets that were conforming which went and grouped with the other data set. But if there was some material that was non-conforming, it's not going to be contained in the PCA ellipse that you saw earlier. We demonstrated the model performance at commercial scale, which is an important thing. In continuous manufacturing, most of the times your development scale is your commercial scale. So we can typically use this scale, which is one of the advantages of continuous manufacturing. You avoid tech transfer and scale up. And so one of the things that we still need to do is we need to include all the possible variations. We did not include, for example, the variation in the API particle size, which might affect dissolution. So you need to keep into consideration all these points when you talk about the validation strategy and releasing batches based on NIR data for that matter. And so we had to validate this model using an external naive data set. So the 6 tablets that you saw on the previous graph or the previous slide were basically made on a completely different day. We started the machine or the continuous line from scratch, and then we made those 6 tablets. So those tablets were made on a different day at a different time by different people. So it was a completely naive data set. And so I will conclude my talk by saying that in this case, this is just one example. Let me reiterate that this is just one way in which you can predict dissolution. There are multiple different ways out there. But we just addressed the real-time release challenge in continuous manufacturing for tablet dissolution for this formulation using this strategy. And so we used a quality by design approach since we tried to understand what was happening with the process and how that affected dissolution. We could predict dissolution profiles for individual tablets using just their NIR spectra, which is an important thing, prediction of dissolution profiles for individual tablets. This is a fast prediction method. It circumvents the need to have process parameters and need to have a model for every dissolution time point. There is a different approach where people, what they have done is basically have a dissolution model for regression model for every data point, and it has a lot of negative points to it. And so we have examined both model dependent and model independent approaches here. And there's a lot of future work that could be done here. We need to investigate more complex formulations as one of the important things. So with that, these are the publications out of the work, conference proceedings. And then I would like to thank the team here, my advisor Fernando Muzio, my co-advisor Alberto Coutinho, and Ifan, Goldshade, and Gerardo, without whom I could not have done this work. Ifan was the one who had done the model independent fittings for the dissolution data. And then NSF Engineering Research Center at Rutgers, and the AIR grant for their monetary support. So with that, I'll give it back to Sri, and then I can take questions.
Thank you very much, Dr. Pawar. So I'm going to begin the question and answer session now. So the first question, are there any situations where we cannot use NIR for dissolution prediction?
Thanks, Sri. So absolutely, there are a lot of situations where you cannot use NIR. And so you have to have an alternate way in which you can build your models. So one such example is where if you have API concentration in your tablets, which is way below the detection limit of NIR. So say you have 0.5% API in your tablets, or 1% API in your tablets. That could be way below the NIR detection limits. So you cannot determine the dissolution profiles using NIR. And that's when what you can depend on is the process data. So, and people have done that. They've used the process data, meaning is my feeder feeding the correct amount of API? Is my blender blending at the right speed? What is the residence time from my feeders to my tablet press? And looking at all these things, you can correlate your, as I said earlier, your process parameters, your material attributes, and your in-process data, if at all any. And you can have a dissolution model based on all these things instead of using NIR data.
Great. Next question, did you use offline discontinuous or inline continuous measurement, NIR? That's one of the questions.
So I would say we've used at-line NIR measurements, which is where you divert the NIR stream that's coming out of the tablet press, and it takes around 15 seconds to scan a tablet or even less. And we've not used an inline NIR for tablet monitoring only because of the lack of the availability of technology. If we are made available the technology to scan NIR inline, then we very well could use it in this framework that I mentioned. People are trying to do measurements like blend uniformity measurement in the feed frame, and they're using that to correlate it to dissolution. So that could be a possible inline NIR measurement which could be used to predict dissolution of the tablets. So I've seen people use NIR in feed frame, so that could be a possible inline measurement. But for tablets, we do not have any as of now, as per my knowledge.
Okay, thank you very much. Next question, what are the challenges you foresee for BCS2 or for compounds?
So I would say your dissolution profiles, I haven't seen any particular example of this right now, but I would say the dissolution profiles should be, or your dissolution method should be able to distinguish between the different dissolution profiles, meaning, let me put it in a simpler way. So you should be able to see the effects of your process parameters and of your material attributes on your dissolution profiles, because for building a calibration model, you need a lot of variability. So maybe that could be a probable answer to that, but I could go back and look on this.
Okay, thank you very much. The next question will be our final question. Which factors or responses can be considerable in QBD if NIR is selected for characterization?
So I'm trying to understand the question. So which factors or responses can be considerable in QBD? So anything that the NIR can pick up is an important thing in QBD. So for a process, for using NIR for characterization, you have to make sure you're not just sticking your NIR everywhere and just taking any measurements that you want. You have to make sure you understand the principles that you're using the NIR, the reason you have to have a sound logic while you're using your NIR. So for example, you cannot use NIR for a random measurement. You can use NIR for concentration measurements, for example. So that could be one of the factors that you need to consider while you choose your NIR for different measurements. You cannot use NIR if your concentration is way below the detection limit, if your API concentration is way below the detection limit. Okay. If that answers it.
Okay, thank you very much. So that's all the questions that we have today. I would like to take this opportunity to thank our presenter, Dr. Pallavi Pawar, for sharing her knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com/webinars. Thank you very much, everyone.
Thank you.